It really is my pleasure to be here today. And as I was walking up the stairs, now that I'm standing up here, all kinds of emotions and feelings are actually bubbling up a little bit. I feel like I'm looking at the future of our discipline. And it's making me proud. It's making me excited that our future of mathematics looks like all of you. It's giving me hope, giving me light. Love it. All right, so in this talk, what I'd like to do is deliver it in 3 parts, if you'll bear with me. The first part, I want to just give you some general background about mathematical biology, which is the overarching discipline that I work in. I want to take you through some of the history of how math has intersected with biology and has had lasting impacts. I'll talk to you about a variety of different examples of mathematical biology in different areas that are going on today in the 21st century. Then in part 2, I'd like to talk to you a little bit about my specific research in mathematical oncology. And in the last part, because this conference is about meeting people and getting to know people, I want to tell you just a little bit more about me. So I'll do that at the end. All right, part 1. Milestones throughout history where math and biology have kind of collided or mathematical thinking has intertwined with some biological thought. Now, I went to try to find the oldest example that I could come up with, and the oldest example I could find was the Fibonacci sequence. Everybody knows the Fibonacci sequence, right? It's the one where you get the next number in the sequence by adding the previous 2. Famous. Well, the original problem that Leonardo Fibonacci was interested in studying had to do with population growth under ideal circumstances. So his little thought experiment was, say I put 2 newborn rabbits into a field, 1 male, 1 female. At the end of the first month, they mate, and at the end of the second month, the female reproduces another male-female ideal pair. His whole scenario was ideal, so these rabbits never died, and that same reproductive cycle repeated. And his question was, how many pairs of rabbits would I have at the end of a year? And lo and behold, you get the Fibonacci sequence of numbers by thinking about things that way. Today, Fibonacci numbers are considered nature's numbering system. So when people tell me, I don't see math every day in everyday life, I don't use math in everyday life, I say math is all around you, and I really mean that. Fibonacci numbers are all around us, right? Everywhere in nature, from leaf arrangements on plants, from the florets on flowers, bracts of a pine cone, scales of a pineapple, all Fibonacci. Right? So this was the oldest example back in 1202 that I could find of how thinking about natural systems led to interesting mathematics that we still use and is very, very, very relevant still today. Now moving ahead several hundreds of years, from Fibonacci, which is a sequence of numbers, let's move to a system of dynamic equations. So the interaction I want to talk about here are a famous set of equations that model predator-prey interactions. So has anyone heard of the Lotkavolterra equations? So I see some nodding. So this is a famous set of equations developed by an Italian mathematician and an American biophysicist. And they were looking at how species interact in nature. And like Fibonacci, they pretty much modeled an ideal situation, really restrictive assumptions. They said, what if there's a predator that relies on a single prey species as its food source? And what if the prey had unlimited natural resources for food and only had one threat to its existence, and that's the predator? If you translate those words into simple mathematical expressions for the rate of change of the predator and the rate of change of the prey, turns out you get oscillations, oscillatory behavior. Now, when they discovered this, they didn't know for sure if natural species behave that way. So there's a very famous data set on predator-prey interaction that kind of had these ideal characteristics. This is the Canadian lynx and snowshoe hare data from the Hudson Bay Company. So they were a pelt or fur trading company that kept records of both the lynx and the rabbit or the hare for almost 100 years. And they definitely saw these oscillatory patterns. So of course, this model, developed in the 1920s, has been iterated on, it's been modified, extended in all kinds of interesting ways. Today, with the computing power that we have, mathematical ecology students are able to go out in the field, collect spatiotemporal data for where species are in the field and how they interact, implement this kind of modified Lotkavolterra model in space and time, and watch all these interesting complex dynamics emerge. So again, an example of how thinking about biological systems has led to interesting mathematics that's still having an impact on how people think about these kinds of things today. The next example I want to give you comes from neuroscience. So neurons, of course, are the electrically excitable cells of the nervous system. And they communicate by propagating what are called action potentials. These are sort of electrical signals that are generated by the excitable cell membrane of the neuron. So a few decades later, 1952, Alan Hodgkin and Andrew Huxley introduced the first mathematical model that explained the mechanisms that underlie the generation of these action potentials. And so they basically wrote down a conservation equation. They wanted to know what an equation for the total current flowing through the cell membrane. In order to write down an equation for that, they had to make some assumptions. They said, well, what if the cell membrane acts as a capacitor that could take on charge? And then they had to recognize that also there are means for current to flow out of the membrane through ion channels. They did experiments and determined which ion channels were most important. Turned out it was sodium and potassium. Put all of that into their mathematical model. And lo and behold, the solution of their mathematical model was an action potential, a spike in energy, right? So, and it had a particular shape. There was a resting phase. Here's the generation of the signal. And then the relaxation of the signal falls below the steady state and then eventually comes back to rest. So this action potential generated by this Hodgkin and Huxley model matched what they were seeing experimentally. Remember, this is before computers. This is before the electron microscope. They got all of this information from their model in that setting, and they won a Nobel Prize for the work in 1963. Even more than that, they sparked interest in an entire new area of mathematics called excitable systems. Even more than that, these equations are still foundational to computational neuroscience today. The last example from the historical point of view I want to give you comes from looking at what we see in nature. What we see like animal coat markings, what we see in terms of patterns on seashells, things like that. So in 1952, a British mathematician, Alan Turing, published a set of equations that tried to explain some of these patterns that we see in nature. The interesting thing about these equations was that you could describe the way in which these non-uniform patterns, so this giraffe clearly is not one solid color. There's big patches of color, right? So that's a pattern, that's non-uniformity. The equations he wrote down showed how you can get this type of non-uniformity, patches, stripes, spots, out of a completely homogeneous, uniform state. So he proposed, for this particular example, that the pigment patterns that we see on adult animals are laid down early in gestation, and they might be the result of two chemicals interacting together that spread out through the embryo at different rates, right? He supposed that one of these chemicals is an activator of some pigment characteristic, and one chemical is an inhibitor of that same characteristic. And that inhibitor might kick in periodically to shut down the activator. If that were to happen, he could imagine that maybe you could get all of these kinds of different patterns. And it turns out that if you translate those assumptions into a system of equations, so remember first we just started out with a sequence of numbers, Fibonacci, then we moved into systems of ordinary differential equations with the Predator-Prey and with Hodgkin-Huxley. Now, if you translate these assumptions, which have to do with both time and space, into a mathematical representation, you'll get a partial differential equation. So he has partial differential equations that describe the rate of change of an activator and inhibitor, how they react with each other, and how they diffuse or spread in space. And it turns out you solve this system of equations on different geometries and on different domains, and you can get almost all of the kinds of patterns we see naturally, especially animal coat markings, seashells, fish, all kinds of things. An interesting thing that he found was if you solve these equations like on a tapering domain, like this, like a tail of an animal that tapers, you can get stripes, you can get spots that turn to stripes, but you'll never get stripes turning to spots, and there's a mathematical reasoning behind that, and you never see that in nature when you look. His models are so detailed that you can actually get these patches on giraffes. The models can predict, look, you see how these stripes on the zebra go this way, and then right at this juncture, there's like a little triangular region. The model is so good that you can actually predict these stripes on the leg coming up to, that are horizontal, turning into these vertical stripes on the body. So this one mechanism could generate all of these interesting things. So these are some examples of mathematics that happened, that was studied long ago, that still has implications for today. So now let's look at a few of the things that are happening now with mathematical biology. A lot of it still takes insight from some of the things that happened in the past, but we're doing different things with mathematical biology today. So the first example in this section I want to talk about are models of global epidemics. Pretty timely topic right now, but does anybody remember SARS? Anybody remember SARS? So that disease kind of scared us, right? So that sort of spread so rapidly, showed us we are really vulnerable to emerging infectious diseases like this. It also did something else. It opened our eyes to the importance of travel. We as a people travel from place to place, and the travel routes we take are vectors for transmitting these diseases. So a model based on actual travel and census data from over 3,000 urban areas in 220 countries was developed by some mathematicians in Italy and France that accurately could predict the SARS pandemic. So this was after the pandemic happened. They went and got all the data on the routes that the virus could spread from Hong Kong. And so the thickness of these arrows represents the probabilities they put on the possibility of that route transmitting the disease. And then what you're looking at up here is a comparison of the mathematical model predictions of an outbreak compared to the World Health Organization's data for whether an outbreak occurred or not. So solid red means the mathematical model correctly predicted an outbreak in all of these regions. The striped countries are where the mathematical model correctly predicted that there was not an outbreak of SARS. And you see some green, those are the regions where the model and the data did not agree. So for most of the world, the mathematical model and the data actually did a very good job in capturing that data. So this research highlights how if you can be accurate in predicting the spreading pattern of an epidemic, you have to be able to connect that to identifiable routes at which routes for how that disease was spread. And I think, like I said at the beginning, this is particularly timely with the coronavirus looming. And so these kinds of models that already exist now can be used and applied to data for these new emerging infectious diseases. Another example, this is one of my favorite examples, that's not my own research, is using mathematics to help predict successful surgeries. So in this example, mathematical modeling and patient-specific imaging are used to build a computerized model of a person's heart and their vasculature in order to see how blood flows through that particular architecture. So you can take several images of a particular individual's heart, put that into the computer, have mathematical equations that describe the blood flow through those geometries, and then you could see what happens on the computer if you give a shunt or you do some sort of surgical intervention before you ever do it on a patient. So this is the kind of impact that mathematics combined with biology and medicine can have. And this is one of the things I'm most excited about, I think, is the possibility of mathematics playing a role in personalized, precision, targeted medicine. And this is a nice kind of segue into part 2 of my talk, which is on my own research. So I'm also in the field of mathematical medicine, if you will. Probably mathematical oncology is the kind of the term that we're using nowadays to describe what I do. So Time magazine, a couple years ago, put out the front page of Time magazine looked like this: How to Cure Cancer. You flipped over the magazine to read the article, and this was what you saw. The hero scientist who defeats cancer will likely never exist. And then they went on to describe cancer in this way. They said it is an intricate and potentially lethal collaboration of genes gone awry, of growth inhibitors gone missing, of hormones and epigenomes changing and rogue cells breaking free. It works as one great armed force attacking by the equivalent of air, land, and sea in stealth. And we think we're going to take it out with what? A lab-coated sniper? Kind of funny ending, but some pointed sort of insights behind some of what's written here. Cancer is such a complex disease. How are we going to cure it? Will there be one person who can do that? Time magazine said no, there will not be one person that will do that. That will never happen. What they did say is there won't be one hero, but there will be many heroes. And I believe this. It's going to take a team-based, cross-disciplinary approach to cancer research. That's what's going to upend tradition and deliver life-saving results faster. So I know this is a little hard to see, but in the Time magazine article, they mentioned all of the usual suspects. They said you have to have biological scientists, you have to have oncologists. They included biomedical engineers, informaticists, and clearly this writer did not know me because he forgot to put mathematical modelers and mathematicians in the article. So I had to draw us in right here. There. Now I feel better. No, and I don't mean me personally. What I mean is the contribution of a collective group of people bringing mathematics to bear on this problem. And I think we who try to do that will eventually be an integral part of what happens in the future of cancer. So I'm going to tell you a little bit about cancer and a little bit about a particular example of kind of work I do now. So things are going to get just a little more technical for a little bit, so just bear with me. All right, first, before I can tell you about models that we develop, I just want to remind you a few things about cancer and why it's so complex and why the models that we develop have to sort of be equally complex. First of all, as you know, cancer starts off as a mutation in your cell, right, in your normal cells. They acquire a series of mutations that transform them, and they stop listening to the surrounding cells. When they get signals to stop dividing, they don't listen. When they get signals that say it's time for you to die, they don't listen. When they get signals that say you need to stay here and do your job, they don't listen, right? And eventually a tumor forms. Now this tumor that forms is a small mass, maybe a couple millimeters in diameter, that's getting its nutrients from your existing blood vessel structure, your existing vasculature. So tumor cells don't like that because not all of the cells are getting adequate nutrient. So they initiate a process called angiogenesis, which is the formation of a new vascular network from an existing supply. So they basically trick your body's blood vessels into making new blood vessels just to feed the tumor. And when that happens, now they have a highway that connects those cells to everywhere else in your body. Those cancer cells can enter the bloodstream, enter the blood vessels, travel to new parts of the body, and start new tumors forming in a process called metastasis. So these are the four big things, four big progression steps in in solid tumor growth. And when you think about this, each one of those progression steps, lots and lots of stuff has to go on. We talk about from proteins and signaling networks to single cells making wrong decisions, telling other cells to make wrong decisions, and then you get this whole population of cells doing the wrong thing, right? What's interesting is that mathematical modelers have been interested in answering questions at each of these levels, have been interested in developing models at each of those levels or each of those biological scales. And if you look over here, there's all kinds of different types of mathematics and types of modeling that has been used. And you pick the type of mathematics that kind of fits the question, the biological question you're trying to answer. And you also notice that some of these are shaded and a little blurred, and that's on purpose as well. Because now, in today's modeling community, people aren't just using one approach. People aren't just using ODEs or aren't just using PDEs. They're mixing these approaches in order to answer questions that cut across biological scales. So you might have to combine mathematical approaches if you want to do that. So the particular biological scales that I work on are, I look at signaling networks and how those feed into single cells making the choices that they make, and how those single cells lead to the population level behavior that we see when we measure a growing tumor or a shrinking tumor if we're giving it therapy. And I've used almost all of these mathematical approaches at one point. The types of models I'm going to talk to you today about use ordinary differential equations. All right, so I'm going to get into a specific example for what I do. My work has always had an eye towards molecular therapeutics or targeted cancer therapeutics. So when you think of cancer therapy, you probably think of surgery first, and then maybe the next thing you might think of is chemotherapy, and that it has been the hallmark for medical treatment of cancer for decades. But we also know that these are drugs that target rapidly dividing cells, including cancer cells, but not limited to cancer cells, right? So certain normal cells are also affected by these drugs. Targeted therapies are drugs that are designed to be more specific than that. They interfere with either specific cells, enzymes, or proteins that are necessary for a particular type of cancer to grow. So I want to give you an example of a therapy that targets a particular type of cell and show you how we kind of developed a model for that. So the primary goal of these therapies is kind of what I said I was so excited about for the future of mathematical medicine. It's to fight cancer with more precision, with more patient-specific information and knowledge. And then we should hopefully have fewer side effects and better results overall. All right, so I want to talk to you about a particular targeted therapy. We're targeting a particular type of cell, and the type of cell we're targeting is called a cancer stem cell. So what is that? A cancer stem cell is a type of cell that has been identified as a driver of tumor growth in a variety of different types of cancer types. Few of their features, they're always a small population of cells. They're the minority. They're not the bulk of the tumor. They're a very small, often hidden minority of cells, but they drive the entire tumor's growth. They divide and self-renew, make more of themselves unlimitedly. They are tumorigenic, which means they are the ones who can differentiate into all the other cell types and create new cancer cells. They're also, of course, the hardest to treat. They're resilient. They're often resistant to the drugs that are traditionally given as chemotherapy. They can be resistant to radiation. And any other kinds of stressors that you try to put on them, they can resist all of these things. So we want to target this population because tumors that result from these cancer stem cells, I'm going to call them CSCs, tumors that result from them have this heterogeneous mix of all different kinds of cells. So we might have a small portion of these stem cells, but then you have a whole bunch of other kinds of cells making up the bulk of the tissue. So we want to target this small population of cells that we think are the drivers. Why do we want to do that? Because traditional therapy doesn't do that, right? So a traditional therapy, you have all of these non-stem cells, and you give the drug, it kills all of these cells. So you get a really huge reduction in tumor. volume, right? The number of cells goes down. You may think you're in remission, you may think everything looks good, but you've left behind the small population of dangerous cells. Eventually, at some point, those cells have the ability to regrow the entire tumor. The new targeted approach would leave all of the bulk of the cells alone, but target that driver cell, target the cancer stem cells. So you would not see a huge reduction in the tumor size after treatment at all. It would take months and months and months for the tumor to shrink down to nothing, but when it's gone, it should be gone because this driver cells have been eliminated. That's the ideal situation, right? So we want to think about maybe combining traditional therapies, chemotherapies, with these new targeted therapies that should come together and attack the tumor from both directions and hopefully give better results. Just a few more biology slides, bear with me, we're going to get to some math. So what chemotherapeutic drug are we going to use? We're looking at a drug, a common chemotherapeutic agent, and we wanted to see how it impacts the driver cells, the cancer stem cells, over time. And what we saw with experiments is that if you give the traditional chemotherapy, you actually increase the amount of stem cells in the remaining tumor. And you also see that, as we said, regrowth and spreading occurs over 50% of the time. So traditional chemotherapy, experiments bear it out, are not good for the driver cells. You're leaving them behind. They're primed and ready to create new tumors. If you give a targeted therapy, right? If you give a targeted therapy, I'm going to call our targeted therapy TCZ, the number of stem cells goes down after therapy, as it should. So that's the idea. The motivation and goals. We know that the standard conventional therapy, the chemotherapy, and the targeted therapy work in opposing directions. Chemotherapy gives you more stem cells, targeted therapy gives you less. So they're opposing each other. So we want to use mathematical modeling to propose a rational schedule for combining these two things so that maybe so conventional therapy, you're going to get relapse. Targeted therapy, you're going to get slow regression. But combination therapy, maybe you can actually get eradication. But how? How are we going to give this therapy and make this work? How are these drugs going to work together? So that's one of the questions that we've been interested in. So we've developed a modeling framework, I'm not going to go through all of this, that kind of helps to address these questions. It's what's called a multiscale mathematical model, in that we look at things that happen at the cellular level. So we would have our stem cells. We said they can differentiate into all kinds of other cells. So we have those cells that make up the bulk of the tumor as well. And then we have the key target that helps these stem cells grow. And we modeled the molecular details of the drug that we were going to give to target the tumor. And so we write down a full system of mathematical equations. It's actually a pretty big system of mathematical equations. They're all ordinary differential equations to kind of model both the tumor's growth and its reduction to both types of therapy. Now, what's important to know is once you have a system of equations, that's all you have, system of equations. You have to connect that to reality with data. So I have an experimental collaboration team that cranks out a lot of data for us to use to train and calibrate and connect our mathematical model to real cancer research data. So this is just a picture of some of the kinds of data that we're able to use to train the model before we're ever ready to make any prediction. We have data about the traditional chemotherapeutic drug. We have data about how the cancer grows under different situations with the stem cells, and we use all of that information for training and calibration. All right, so here's some of the kinds of results that we can get. So the first thing we wanted to look at is, can we compare our mathematical model, its output, to data for how a targeted therapy might work. So here is the tumor volume after treatment with a targeted therapy. This is the days after implantation. So the black line is no treatment, right? So that's black line is no treatment. And then the two green curves represent two different dosing schedules, one the drug given on 2 different days. And what you see, the solid lines are the mathematical model predictions, and then the data points are the circles and squares. So, and this is not parameter fitting, this is a direct comparison, just laying our mathematical model on top of the data, and we got pretty good results. Now, if you look at the amount of stem cells, or the percent of the bad driver cells in the body after the targeted therapy, you see that you get a significant reduction in the stem cell pool, which is exactly what you'd hope to get. And this is after 60 days. Now, if you look to see what happens if you just give traditional therapy, first thing you notice is that you don't reduce the tumor hardly at all compared to what you did with the targeted therapy. So here's the tumor volume again, and then these are our mathematical predictions. But the thing to note is that the stem cell pool, right? So without treatment, here's what we would get. With treatment, we get huge jumps in the stem cell number, right? So this drug is doing the exact wrong thing as we expected it to in terms of the stem cell pool. So what if you give combination therapy? Here's our mathematical model predictions. Again, in the blue solid lines are our model predictions and the data for what happens if you give combination therapy. Now we see that you can counteract the effects of just giving the traditional therapy alone, and again, you're getting that big reduction in the stem cell pool. Okay, so this is all well and good. We like that our model seems to be doing okay. It seems to be matching the data. It seems to be generating the same sort of results that they see experimentally. But we really haven't said anything about how do we give these two drugs together to make them work well. So that's what we really want. That was the real goal of this. How do we give these two drugs together? So if the goal is to optimize therapy, we have to figure out what we mean by saying that. So we want to minimize the amount of drug that we're giving, the amount of the targeted therapy we're giving. And at the same time, we want to maximize the amount of stem cell reduction. We want to make that stem cell pool as small as possible, giving the smallest amount of drug as possible, right? We might be able to change to accomplish this by changing when and how often we administer the two drugs and how we give them in relation to each other. So that's what we wanted to see if that made a difference. So we had to test some dosing schedules. So this is a traditional dosing schedule that my experimental collaborators used. They give the targeted therapy either for 3, 4, or 5 weeks. And then the chemotherapy is given either before, at the same time, or after the targeted therapy, only for 2 or 3 weeks. All right, so now we have a dosing schedule to play around with. We can play around with all of these things. But we need some metrics. So we need a metric for how much drug we're using and how it's generating tumor cell reduction. So we have the amount of targeted drug required to reduce the tumor. We just picked 70% by the end of 6 weeks or 8 weeks. Right? So that's our metric for the amount of targeted therapy and how it's reducing the tumor. We need a metric for synergy. Are these drugs working well together? So we have a dosing synergy metric. If this particular metric is greater than 1, the drugs are antagonistic. If it's less than 1, the drugs are behaving synergistically. And then we need some measure of the percentage of the stem cell reduction. So remember, we want those bad cells to be as small as possible. So those are the 3 things we're going to look at. We're going to run simulations of this model hundreds of times for different dosing strategies and figure out when do we get synergy, when do we get a small amount of stem cells, and when do we get a huge reduction in tumor volume. So here's an example of just a couple of these runs. So here's an example of 35 different runs. Just to walk you through this, remember we said we were going to give the targeted therapy for 3, 4, or 5 weeks. The shaded gray regions means we're giving targeted therapy. So that's 3 weeks, this is 4 weeks, this is 5 weeks. And then we said we're going to give chemotherapy, which we called CIS, for 3 weeks or 2 weeks. And so we played around with it. The key thing I want to point out here is this middle column. Remember we said we had an index that told us whether the drugs are behaving synergistically or not. In all of these simulations, the drug the drugs were not working well together at all. Even though at some points we're getting large amounts of stem cell reduction, we're not getting any synergistic attributes of these drugs together. So these are the dosing strategies my colleagues, my experimental colleagues wanted to look at. We decided to look at some non-conventional dosing schedules. And we started spacing out the drugs, right? So leaving some space, and immediately we began to get synergy. And we could even improve the synergy by doing different kind of spacings of the drug, by decoupling when we gave it. So this is all giving the drugs, both drugs at the same time. Look what happens if you give targeted, followed by chemo, targeted chemo, targeted chemo. You get the best synergistic results. And you're still reducing the stem cell pool, and you're giving very little targeted therapy. So spacing out doses is what the model predicts will be the best avenue for leading to synergy between the two drugs. So since this is what the model predicts, let me summarize real quick and then I'll tell you. So this is one example of the kind of work that we do. We try to develop versatile mathematical modeling frameworks that operate at the levels of the biology that we understand, intracellular, cellular, and tissue levels. We want to develop these models in a way that they're able to capture the real mechanism of action of these new drugs that they're testing out. Most of them are targeted in nature. And they're able to be used to investigate the impact of the cellular properties of tumor growth and reduction, and they're able to say something about how you should deliver these drugs together. So what's next? Well, we have a prediction. We said there's a particular strategy of maybe pre-treating with targeted therapy followed by chemo and mixing that up that worked the best. So my experimental collaborators are generating tons of mice, and they're going to test our mathematical modeling strategy. So this is the validation step. So when you think about the modeling process, we had to figure out a biological problem. We had to be able to articulate that problem in words. We had to be able to translate those words into mathematical equations. We had to be able to input data, real data, into that model in order to connect it to reality. And then we had to make predictions. But we can't stop there. We have to validate those predictions, and that's the next step that's happening now. They're going to be testing out the dosing strategy that we predicted on all these mice to see if we were right. And then, remember when we talked about some of the milestones and stages through history, we started talking about ODE models, the ordinary differential equations, and then I showed you an example of partial differential equations that incorporate space. The model that I just talked about did not incorporate spatial features of tumor growth. It only incorporated the temporal dynamics of drug delivery and tumor reduction. So another next step would be to develop a spatiotemporal model of these events, which will add in additional information about where and how cells move, and that may impact how much drug they receive and all of those things. So that's one of the next steps too. All right, so that's the end of part 2. Part 3, I don't want you to walk away from here without knowing a little bit about me. I hope I get a chance to personally talk to every single one of you, but in case I don't, I want you to know who you saw today. Who is this person, right? Well, that was me. I was just a regular little girl with a regular little family. But then somehow I became that. I was so cute when I was little. Somehow I became that, right? I became an applied mathematician, a theoretical biologist, a mathematical oncologist. How did all of that happen? Sometimes I don't even know how it all happened. But I can tell you something that really put me on this path. Probably the first thing that put me on this path. Besides my family, my parents, big proponents of education. My dad would sit down. I had an older sister, 2 years older than I was. So when she was getting ready for kindergarten at 4 or 5, I was 2 or 3. He made me do everything she was doing. So we both learned math and science and all of those things at home before we ever went to school. So I had a family who really valued education, which was great. But then I found a cohort of students in high school because I attended high school, we called it math camp. It was the math and science honors program at Arizona State University where I lived. And so the summer after my junior year, I spent 8 weeks on campus taking a calculus class at ASU with a bunch of other underrepresented minority students. It was a program to get underrepresented minority students interested in mathematics and the sciences. And I had gone to a predominantly white school, so going to this program and finding all of these people who looked more like I looked, liking math, it was an eye-opener for me. It was wonderful. So I found this cohort of students, and I met the director, the founder of this program, and it really, really changed my life. It really did. It's the reason I decided to go to Arizona State University for my undergraduate degree, and it's pretty much the reason I became a math major. A life-changing moment. I went to college going to be an engineer because that's what you did if you were good at math, right? You just kind of go to college and you become an engineer. So that's what I declared as my major. And I walked into my engineering classes in the engineering buildings, and I did not like it at all. I knew immediately that that was the wrong choice for me, but I didn't know what to do about it. Luckily, I had gone to this program for 2 summers. I knew some professors, I knew some people, and I was taking a class with the director of the program, a math class. And one day after class, he said, "Come to office hours." I didn't know what office hours were, really, and it certainly sounded bad the way he ordered me to do it. I thought I was in trouble. I thought, did I mess up on my homework? What happened? And so I go to his office, and I'm terrified. I'm literally shaking in my boots. I don't know if he's going to make me do math on the board. I don't know what's about to happen. And he asked me what my major was, and I told him, I'm majoring in engineering. He's like, "Why?" And I said, "Because that's what I'm supposed to be doing, because I'm good at math." He's like, "That's not what you're supposed to be doing." And so I said, "What am I supposed to be doing?" Because I really didn't know. And he said, "You should think about being a math major." And those words sound really simple and easy, but to me, it was like an invitation to join this group of people who I thought were brilliant and special. And he said I should think about being a math major. And those words set me on the course that started this whole mathematical, applied mathematician movement in my life. I can't imagine where I would be if a professor hadn't taken the time to show interest in me and in my ability and to invite me to join the discipline. It was a real life-changing moment. So I was a math major now. I was a pure math major, so I was going down a very pure math track. And I was enjoying it. I didn't know anything else. I didn't know anything better. But when I was in college, mathematics and biology were still very separate. It wasn't like it is today where people are kind of appreciating math and biology a little bit more. So I didn't think I hadn't taken a biology course probably since junior year of high school. Biology was not on my mind at all. But then I'm walking through the halls of the math department, and I see a flyer that had this picture on it. And I thought, why is this in the math department? What is this about? Right? Spots, leopards, has nothing to do with mathematics. And I did something I'd never done before. I went to the talk that this was advertising. There's seminars and all kinds of things going on in the department. I never went. I'm an undergraduate. I'm not going to understand anything. Something prompted me to go listen to this talk. So I go to this talk. It's all over my head. I don't know what he's saying. I don't know what math he's doing. But what I do know, what I do walk away with, mathematics helped answer questions about something in developmental biology. That just blew my mind wide open. I'm like, you're kidding. You can do this? And so at that point, I knew I wanted to try to use math in that way, in some way. to answer scientific questions. And it was a long uphill battle for me because, like I said, I hadn't taken much science, especially biology, in a number of years. But I decided I wanted to go to graduate school. I wanted to work with this guy who gave this talk. And so I applied to the graduate program in applied mathematics at the University of Washington, and in hopes of working with James Murray, who was the guy who gave this talk. And for some reason, he accepted me as a student, and sort of that put me on the road to mathematical biology and mathematical oncology. So I've had an interesting yellow brick road, windy road that's gotten me here today. As you know, I started off as an undergraduate at Arizona State University. I went to the University of Washington in Seattle for my PhD, amazing department in applied math there. And then I went to the University of Minnesota. So I graduated in 1990 or something. And that year, they happened to have the very first theme year in mathematical biology at the Institute of Mathematics and its Applications, which is at the University of Minnesota. So I spent a year in residence where I got to meet every person whose paper I read. I was starstruck, awestruck. Everybody who was anybody in this field came through that year. And so I got to build a network of people, get to know people, and that was a really foundational year in my career as well. Then I went to Duke University for a second postdoc, where I also had great mentorship and great projects to work on, and another great time in my life. So when I look back on how I turned from that little girl to what I am now, it really took a village of different people to get me there. So this is Dr. Joaquin Bustos. He's the one who started that math program at Arizona State University for underrepresented minority students. He's the one who said, come to office hours. So I owe him a lot. This is James Murray. He was my PhD advisor. He put together that flyer that got me to go to a seminar and got me working in math biology. This is my collaborator for my PhD work who spoiled me rotten, and I thought, hey, this is easy. I just go, I find a collaborator, they do all the experiments I want, and it'll be great. And it doesn't work like that, but he was great. So Mike Reed and Arlie Petters were my mentors at Duke University, and then a whole host of people who have shaped my educational activities, the kind of educator I want to be, are all these people up here. And you'll notice, so this is Sylvia Bozeman. I don't know if anybody knows Sylvia Bozeman. She's at Spelman. Not all of them look like me. Only one of them is a woman. But they all were instrumental in helping to make me what I am today. So it took a village, and I'm so appreciative of all of these people. And I guess the moral of that story is kind of go to office hours and keep your math major, go to seminars. You'll figure out what you want to do. Anyway, thank you for your attention and your time.
Does anyone have a question they'd like to ask at this time?
Oh, here or there.
Hello, I'm Jessica from Central Washington University, and I was just curious about your scheduling index. What goes into that? How do you determine that?
Yeah, that's a good question. So we look at the amount of drug that it takes to reduce the tumor by 70% when you co-treat. So that's our numerator. And we divide that by the amount of drug it takes to reduce the tumor 70% with any of the other dosing strategies. And so we get this ratio, and if that ratio is bigger than 1, that means you're doing worse than you were, and if the ratio is less than 1, you're doing better, so the drugs are working better together, or the treatment strategy is working better. Great question.
We have time for maybe one more question, if anyone is bold. Oh, well, there are a couple, but you were quick.
Hello, my name is Parmita. I'm also from the University of Michigan. Thank you for your awesome talk. My question is about the pandemic modeling that you were talking about. Do you have any idea, any hypothesis as to why for a handful of countries the model didn't match the data?
That's a very good question. I don't really know why. I think the community felt that the model was actually extremely accurate because there were only a few countries. And I bet the reason has to do with the bottom panel of that picture that showed transmission routes. And they had probabilities associated with whether or not transmission would occur to certain regions. My guess is that maybe they weren't sensitive enough in those probabilities of transmission to certain areas.
Let's thank Dr. Jackson again for an excellent talk.